Day 2 data for the precipitate lab. So let's take a look at what we're going to be doing. We're going to be filtering. I think I'll go to a screen share. And in day 2, we're going to see filtering. Okay. And what do we mean by filtering? I'm going to just go to the data table. We made in day 1 8 test tubes. All right. And in test tube 1, we had 1 milliliter of copper nitrate and 8 milliliters of sodium hydroxide, and then we have this reaction. And then in 2, we have 2 and 7, and so on. All right, so that's what we have. And what we want to do is figure out how much precipitate we have. So we're going to filter the precipitate onto a filter paper. We'll let it dry overnight, or actually over spring break. And then when we come back, we'll be able to weigh the filter paper and the precipitate, subtract out the weight of the empty filter paper, figure out the mass of the precipitate and moles of precipitate. So these calculations we will not be able to do till after break. If you know milliliters of copper nitrate, if you know milliliters, you know liters, and if you know liters, you can calculate moles because you know it's 0.5 molarity. Same thing with sodium hydroxide. There is an observation you can make today, color precipitate and color filtrate, that we'll be able to make as well. All right. So I think that's what we're going to be doing today. And I am going to look at these instructions, but I think I'm going to break off the screen share and show you this so you can see this right here. Okay, looking here, we should have data, and let's see, find the glasses here. And we have 8 test tubes filled up. I only show 1. But this is test tube 1. You can see the precipitate is sunk to the bottom, and we can see that it's there. So you can look at all 8 test tubes. I'm only doing test tube 1 as this demo model, but you'd have those other ones there. Okay, now I need to filter this, and what I want to do is collect the solid in there. So I'm going to grab a filter paper. Okay, so I'm going to grab a filter paper and fold it in half, and fold it in half again. Okay, door number 1 is a good option. Door number 2, not so good. It'll just go through. Door number 3 is a good option. So you look at that, you get a funnel, take just a touch of distilled water, kind of get this wet around the edges. Sorry, I should have done that here so you can see it. Trying to do that over the sink. I'm going to get a grad cylinder. And what I want to do is get all the solid into this container here. Now I might have to scrape or do things. I think this will come clean pretty well. And I might even give this another mix again before I do this. And then I'm going to do a quick dump in here. I don't want to exceed the capacity of this, so I'm going to be careful. That's fine. This came out almost perfectly. I want to use as much water as needed, but I don't want to use too much because it'll take longer to filter. So I'm going to grab a little distilled water bottle, teeny residue right here. I don't have much, but a little bit extra in there, shake it around, and then I'm going to dump this leftover in here. Okay, and I'm just going to let it drip, drip, drip until it's done. Now notice the color of the filtrate. I don't know if you can see it, but it is clear in test tube 1, and that is expected if you look at the reactants and think about what might be in excess in this test tube and what color would be the excess reactant, because that's what's dripping through is the spectator ions from the product, but also any excess reactant that you have. Okay, and I would let this go, and ultimately what I'm going to do when it's done filtering is I'm going to spread that out and leave it to dry overnight, or in this case over the weekend. And I think I'll just model that here. It's not quite done, but I'm going to model that how it would be going. And what I'm going to do is have I'll probably have a plate or something that's labeled, and then I'm going to have a paper towel that's labeled. And then I'll probably have a tray for your team actually. It's labeled. Find one, and then you can call this on the paper towel test tube 1 and team whatever, period whatever. And I'm going to just take this out, Put this out here. We'll leave it out to dry. Don't worry if you didn't weigh the filter paper today. It's pretty uniform that weight. You can just weigh an empty filter paper. They're pretty standard. And then subtract out the weight of the filter paper from this time to next time. And ultimately, when it's all dry, you can get the mass. But it's pretty wet, and it's probably going to take 48 hours to do that, but it'll certainly dry over break. So that is an example of a product that we have for this lab and doing that step 2. So it's a lot of waiting around filtering, but then you can kind of be planning your write-up during day 2. And then what's going to happen day 3, you're just going to weigh it, and then you can complete your data table and then be answering those questions right there. Sorry to be talking from a distance here. Okay, so that's day 2, and thank you for watching.